Okay, so to kick off, we go to server, our internet access, depending on the one you have. So for our ligands, our substrates, or those identified compounds from our extract, you will now want to source them. So you simply type PubChem, like I mentioned, PubChem, okay, and then you please leave me know that. So you type that, and this will pop up. You simply hit on your words, your PubChem. Is that okay? So we hit on PubChem, something like this will show up. Okay, so when this shows up, aside every other information you have here, you have this search place where you can search. Okay, let's say we are looking for catechol. How many of us know catechol? Now catechol means okay. It's just or quercetin. How many of us know about quercetin? You've heard of quercetin too, right? Yeah, this is just a compound commonly identified in extracts. Okay. If you typed it correctly, you can see a pop down. Okay, trying to tell you any of these words you are looking for. So I'll simply hit on enter. Okay, and then this is the okay, this is the particle supposed to load here. I think it's from the network. Okay, so this is what you have. You simply click on the name, okay, to get that open up. So I'll click on the name, and then when it loads, it's going to appear like this. Okay, as you can see, this is the table of content. Okay, this is the content trying to tell you all that is inside here. Then also, the first is the structure, the name and identifier, chemical and physical properties, and all that, just for this particular compound. Is that okay? So, as you can also see under structure, this is 2D, which I mentioned, and this is what 3D. Some of them as well can have their crystal structures. Okay, but to download this, I'll have to go down. You can, when you're working yours, you can read other informations there. You can see their molecular weight, the date they were uploaded here, and all that. Yeah, as you can see this since 2004 or so. And also a little PubMed. PubMed now is literature. Okay, information about the catechol is a benzodiol comprised of benzene core carrying through this design. Do you get that? Okay, so we can now go to source this. If you move down a bit to under structures, according to our table of content, you see, I don't know why this is not loading fully, but this is supposed to load the 2D, okay? While if you move down, you're supposed to have the 3D, okay? This is the 3D conform. I've mentioned we only work with the 3D conform, okay? So beside the 3D, you can see some renderings, okay? This is just going to tell you displays of this, which doesn't matter. If I click on stick, it will, are we getting that now? So this is of no importance to what we're doing here. So under here you are going to see download coordinates. Is that alright? The coordinate is to now download this. So I'll hit on this, and then options will show which one am I to download from. Is it the SDF, the JSON? You won't see PDB file here. Okay, even though it's a 3D structure, it won't come out as what PDB. Okay, remember after this we're going to our protein, which must be in protein data bank format. So whatever you're working with, the same protein is going to be in PDB. So what do? I'll download this as SDF. Okay, and when I click on SDF, it has downloaded. Okay, this is already my download. Go to this location, I'll find that compound downloaded. This is today. Is that alright? Okay, so I've downloaded the ligand. However, I downloaded this ligand as an SDF file. Is that not so? It's supposed to be a PDB file, right? So you can easily convert from SDF to PDB by either using both online servers, like I've mentioned, or offline tools. This PyMol I just installed for you, you can open this same library in PyMol and then just save it as PDB and you've converted it. Is that right? You can as well do the conversion online here. So what do we do? For time's sake, I'll simply use the online server here by simply typing convert SDF to what? PDB. PDB. Okay, this is it. I'll just hit enter. Okay, if I hit on enter, it will give me different online servers that can help me convert SDF to what? PDB. Okay, so I will scroll down. I usually have a particular server which is this online smile translator. You can as well use Open Babel. There are many of them, like I mentioned. You can see that PyMol I talked about, even Chimera and all that can help you. It's just to save it to a different format. So here we are going to use what? The Calculus online translator. If you as well search convert SDF to PDB, then online smile, it will certainly bring you to this. Is that right? It will bring you to this. So I can go ahead. Yeah, I can go ahead and now click on this online translator, and then when I do, you have a good battery. Okay, no, okay, that's fine. So this is what we have when we open that server, and you can see online smile translator, structure file generator. So under this, you're going to see a choose file. Okay, you're going to see what choose file. Choose is trying to upload that SDF you downloaded, right? So I'll click on choose file, and then it will certainly show, right? Then you can hit on this. You can see it has uploaded. Is that right? It has uploaded it. So what do I do? I'll come to this right end. You see SDF. It's telling me what is the display style you want to use. So I'm going to use okay. And then aside the PDB display style, I'll still come to this side. Normal structure of the benzene can either be Kekule or aromatic. But Kekule is the usual, so I'll leave it in Kekule, it's fine. Then under the extension, is it going to you can see Kekule here, so it's fine. You also have is it a 2D or a 3D coordinates you need? I need it in what 3D. Is that also and I can hit on it translate. Okay. Once that is done, I'll hit on what translate and that will certainly translate it. This will just come up. Okay. And when it comes up, you see click here, it has been changed already. So I'll simply hit on what click here. Click download. Is that alright? So once that is done, I can now go to my downloads. Yeah, look at that here. It will come out as this CLA whatever. So what do I need now? I need to rename this to what? Vertical, right? I know if it's already in PDB format. It's already in PDB format. By default, this is BS Discovery Studio. That is my default software for opening PDB files. So when it comes here, it will certainly change to that. Is that alright? So this is so the next thing I will do here is to open a folder. That is what I usually do. Okay, I will open a folder and I will call this tutorial class folder. The folder. Okay, you just right click. You see new. Yeah, you see what. So I can drag this into here, right? And then I can delete this. I don't need it for anything again. So this is what I have. Okay, so I have my substrate or ligand inside this now, right? get my target, okay, my protein this time. Like we mentioned earlier, proteins, when you talk about target, it could be enzyme which catalyzes reactions, right? It could be receptors, which most times it's just a channel. Is that not true? It could even be RNA. So when you say target protein, it's actually a tautology, right? Just like target receptor. Target, even in write-up, people do it, but it's always pardon. But it's either you use target or you use protein. Is that okay? Especially when you're making presentation, okay, maybe outside Nigeria. Here, they won't know the difference, but outside there, somebody listening to you will know that you're just mixing things up. So,
I have a question.
okay,
What are the offline listing that can be used to convert the
you can use Open Babel, you can use Pimo, you can use Chimera. The conversion is just to save as. You know what we mean by save as. You type something, you go to save as. Under save as, you see different formats, times, file times you can save. So when you go to save as, you just click on file type, change it to PDP. That's all.
those ones you mentioned are offline?
Yeah, they're all of them. You have to.
And also, so for us,
Yeah,
the this thing, you know, it said it should be downloaded from that Pokemon as
yes,
SDF.
yes,
The last I saw that things there like JSON and all
yes,
that.
yes.
So it's not okay to download it then those other ones.
I don't download in JSON and all that. Yeah.
But is there a way, is there a time somebody can download to that just one button?
And not yet. I've not worked with JSON before. Maybe when you are working with some kind of higher level image calculation, they might require. However, in most cases, they don't. Okay, usually it's some kind of designing or something like that. That's where you might see JSON and all that. Is that okay? So go to our PDP to get our what? Our protein. Okay, I told us we are going to be using COX too as well. I think that's the first time I'm using that protein to teach. That's because the protein is a bit unique, and then I think it will help us to explain one or two other things. Most time I just use a simple protein that will not give me stress. Abi? But it's better we use one that will learn more things from. So once you type PDB and hit your enter button, this will come up, right? So I'll simply hit on the home page, which is the RCSB, and then it will load in this form. Okay, this is the protein. This is the home page. Okay, aside many things you can find here is where you can search, right? You can either search with ligand name here, with your protein name, or you search with your what protein ID. The same thing with PubChem. PubChem as well has IDs, right? Every compound uploaded to the server has a PubChem ID. The same thing with protein. Any protein uploaded to any server will have that server's ID. If it's protein data bank, it will have a PDB ID. If it's a Uniport server, it will have a Uniport what ID, of which you can simply enter that and search with, or you type the name of the protein and do what and search. How do I now get the IDs of protein? I've mentioned earlier that when you are trying to investigate or search a particular target for your research, you can get that through some in silico studies that have been done with respect to that effect. True or false? I want to look at anti-diabetic effect of a particular plant. I'll simply type anti-diabetic effect of Moringa oleifera in silico. I'll hit enter. I'm going to see works, published works where INSILCO, in silico. Okay, I'll simply find you have in vitro, you have in vivo, you have in silico, right? All these are, I think, Latin and Greek words. What they mean? In silico is dry land. Okay, in vitro is test tube. In vivo is wet land or animal related data research. Okay, so um, what was I saying before I got to that?
How you
If you
can
want
get
to
the
Okay,
get
how
the
you
ID?
can get the ID. Okay, you get that through that literature. In that published work, you are going to see the protein they made use of. When you go to the methodology of that publication, you are going to see under methodology, molecular docking studies, you are going to see so and so protein, cyclooxygenase 2 protein in brackets, they write the PDB ID. And it was downloaded in its crystallized form from the protein data bank server and prepared or cleaned using this design. That is how we present our in silico publication. So in their methodology, you are going to see the protein they used and the ID they used. Is that all right? Okay, so by that you can simply come here and search. Most time, to me, I prefer searching here with ID than coming to search with respect. Take for instance, I search cyclooxygenase, cyclo oxy, okay, genase 2, right? Okay, something like this. Let's get okay. Let's see if I got that spelling correctly. What it will simply do is that it will bring out lots of cyclooxygenase 2, okay, from different sources, crystallized and uploaded by different researchers at different dates and all that. I get it complex with different um, inhibitors or drugs. Are you getting that? Take for instance now, this is crystal complex of morindase with this. Then if I go down, this is another crystal structure of morindase. This M is murin, okay? Um, the solution structure of this,
Oh,
okay, this is the ID I'm talking about. It's always a four-letter word. Sometimes two alphabets, two numbers. Sometimes three alphabets, one number. But there must always be a number there. Are you getting it? This one now is two number, two alphabet. This three alphabet, one number, and all that. Okay, this is two as well. So these are their names. Then this is RNA. The complex with this. You can see is even giving you even beyond what you did not even it. Are you getting that? So these are you can see structures. However, in most cases, those ones you have upwards will always okay. So something like this now. Okay, you can see this uh, the cyclooxygenase 2 complex with this apical whatever. Then similarly, this another of it in complex with harmaline analog. This could be drugs or inhibitors. Or is that alright? Okay. But this particular one I'm going to make use of, which I can't even find here, was complex with one of the standard drugs, okay, used for this. I've not investigated this to know if these were actually standard drug, but that one was co-crystallized with a well-known standard drug against the same protein. When I say standard drug, I mean drugs that are used. Okay, sir. Drugs that are already approved that are used for drug treatment. Is that alright? Okay, so let me see if I can record that 3Q7. What is that name? Name of that. Okay, I think I
wait,
remember.
for example, now this,
Yes.
let's say this particular one is the one we are looking for. So what
Okay.
are you going to do to
Okay, that is what we want to do now. Okay, so what I'm looking for now is 3Q7D. That is the ID.
okay, so you get the ID of that
Yes.
and come and type it here.
No, if if I open this, it will lead me to that question you want
Okay.
to ask now. So that's why I don't want to explain that there and still come back here again. So if you click on this or you just click on search, it will load and then it will bring up the. This is something you were asking.
Mm.
Uh, However, okay, let me. Uh, okay, in that other one, if you click on this number,
Mm.
in that other one, this page is what will open, which will now show you only that one. In that other one, yes, it will just show you everything pertaining just that single one you clicked. I don't know if you got it. Is it confusing people?
Mm-mm.
Okay, let me go back.
The question now is you've gotten the ID of
Yes,
that one.
yes,
Then you now say for us to get our ID, this is
yes,
because you already know this ID.
yes,
Then you just type that cycle of vigilance. Then it brought out these things.
yes.
For example, I don't know the ID of my this
Okay,
thing.
okay.
Then
And
I
you want to
typed
use this.
uh,
You simply click on this.
okay.
Okay, if you click on this, it will take you to what we just opened now. It will take you to here. Okay. If you use the ID to search, the ID will simply bring you directly to this. However, this is a similar protein to what we have. They are all murine cyclooxygenases.
Mm.
Okay, they are all murine cyclo. You can see this murine. They are all here. So, but no, I'm not sure about this complex, so I can easily make use of my own complex 3D7Q. 3Q. And most times, as you can see, even if I type 7G, you see that there's a protein that have that.
Mm.
Are you getting it? So you just have to be a bit careful. If not, you might end up working with
Something else.
a wrong protein. Yes. And you're already there. Okay, so this is what we have, right? As you can see, this is the protein. Okay, so all this information are with respect to the protein. Okay, let's start with the up here. This is the ID. We've agreed on that, right? And this is the 3D ribbon view. Okay,
Mm.
structure of the protein. This is the PubMed link, or what they call the PDBDOI. If you carry this and search, this is going to expose to you all the write-up attached to this crystallization, how this was crystallized, whatever study, whatever thing you needed to know about the crystallization of this and uploading here. The same information, if you come down a bit, you are going to see an attached PubMed link, okay, with respect to that. Are you getting that? So this will give you just for information sake and all that. Then, of course, classification and all that. You can now see that mutation, it has no mutation, which is fine. Okay, then, in case you couldn't find an ID, okay, like she mentioned, and then you have a choice, a lot of them to make choice from. There are few things I think I mentioned you are to consider as well before choosing your one. Is that okay? You don't just blindly go and pick one. Number one thing you check is the extraction or structural elucidation method. You check the method of elucidation. You can use X-ray diffraction crystallography to elucidate the structure. You can use NMR. You can use electron microscopy. These are the three major. I think now they are using cryo VR. How many of us have heard about cryo VR? Yeah, that one is one of the fast rising um, techniques now in for protein 3D structure elucidation. They are from scratch. I've attended one of their workshops and it was very interesting. Okay, so that is what that will help you to achieve. So first of all, we usually go for the X-ray diffraction method, except it's not in the option. Then you can go for NMR or electron microscopy. Okay, aside going for just the X-ray diffraction method, you are as well going to consider what we call the Armstrong value or the resolution value of the dog. Good afternoon, sir. Welcome. Give battery we talk. Welcome, sir. Thank you, sir. Okay,
Look
so
out for what again?
you look out for the resolution value of the crystallized protein or target. Resolution value is always um, um, represented in Armstrongs. Okay, you see something like this, you have 2.40 Armstrongs. Armstrong tells you about the resolution. And you know what resolution means. The more resoluted by the target, the better, right? Resolution is
The
telling you
more
how
resoluted the what?
the target, the more resoluted the target.
How clear you can see it.
And it talks about aside clear how original the components of the proteins were after resolution, after elucidation.
You take the highest resolution.
No, the smaller
Okay, the smaller.
the smaller the value,
This one is that do you have an
Hmm.
this one here? Is it this
Take the next one.
in all your tab?
Yes. I claim it. Please close the very well. Okay, so I've mentioned that aside the X-ray diffraction method, we take the what? The Armstrong value of the elucidated structure. The smaller the Armstrong value, the higher the resolution. Very, very important. I'm saying something like 4.3 point here. In fact, they will tell you anything from 2.5 upwards, except you don't have anyone lesser than that. Is almost not uh, highly resoluted. The higher the resolution, the more original the components of that structure will be after resolution. Are you getting that? So it's very, very important. Um, and then the lower the resolution value, the lower the Armstrong value, the higher the resolution. Are you getting it? I'm telling you now that the higher the resolution, the more original the structure will appear as. Okay, yeah, you won't find some component of it being lost at the process of elucidation. Okay, so this is what we have. Um, Aside the Armstrong value we mentioned and the X-ray diffraction method. Also, sometimes you might check the source, the organism source. Okay, this you have musculus. Which organism is this? Okay, maybe we'll check that later. But I know it's not a human being, right? Human being, you see Homo sapiens, right? Yeah, human being, you see Homo sapiens instead of musculus here. So that is what this is like. Okay, so if you have checked that and checked that, that is just a few parameters you.
Instead, you should look at the source of the organism. So when you now look at the source of the organism, like what are you looking for there?
You want to see if there are humans, or if you are studying, carrying out a human research, okay, then you look at human sources for the same protein. Remember, proteins, even though they are the same protein, when elucidated from different sources, they still maintain that's called um, what they call it. There's what they call the conserved species or conserved group of that particular protein or target. They will always have a conserved group, but at the end there should be some level of little differences in structure with respect to the different sources. But the units that are responsible for the catalysis of that protein will always be conserved. I don't know if you are getting it. What it means is that this protein in microorganism is the same protein in human, is the same in animal. Are you getting it? Structurally, they are the same, they are similar. And there's this structure function relationship as far as protein is concerned. These structures are similar. Then there is high, in fact, there is an established belief that the functions will be similar as well. Is that clear enough? Okay, so if that has been established, we checked our elucidation method, we checked the Armstrong value, and then the source. Like I said, if you don't have human source, you can go for every other source. They are all the same. But if there is human, you can go for human. Okay, so if that is done, I can come here and download my file. Okay, you can see two download files here. Okay, let me see what this one will always give me. Okay, and then this. Okay, here you might just have few options to download from. This is FASTA sequence. This is the PDB format sequence of this. Okay, FASTA is talking about the amino acid units, okay, of the protein. If you download this, you'll be seeing arginine, this one of that protein. But if you download the PDB format, you are going to be having the structure, not in terms of sequence now. Is that okay? However, where we download, you go to this second. Okay, this is display style. Do you get that? This is display, not download. If you open this, it will just display that. Then if you come to download, this is what you have. Then we are downloading as what? PDB format. Okay, so the second one, this PDB, but in a GZ file. This Z is another extension, not just PDB. Okay, even in full PDF, you can still have it. Okay, so we've downloaded it and it has entered here. Can we see that as 3Q7?
Yeah, they enter.
Is in my downloads. If I go to my downloads now, okay, so I can now move it to the folder, right? So
What if you don't move it to the folder?
you have to move it so that all of them will be in one folder. Because at the end, we are going to copy the directory of this particular folder. Everything we're going to be working here is going to be in one folder. At the end, you will command your docking tool to access this folder and work with. So if you didn't have a folder for it, it will access the entire download, and I'm sure it will never work because there are many of similar folders in that particular download. Okay, so this is what we have. I hope we follow to this extent. I want us to get to somewhere before we now allow us to go and practice. Okay, so the next thing I do, I will open the protein, okay, which I've downloaded here. My own appeared as 1 in bracket. Who can tell me why that 1 in bracket is there?
Primary protein.
This protein,
Primary protein.
yeah, this protein appeared
One.
1 in bracket. What does it mean? Hmm?
So you can
You know that if you download the same of another one, it will be
So this
First
one,
download.
this one now that is showing 1, what does it mean?
First download.
It's a lie. It means I already have a download of this. Then I now downloaded it again. It will appear as 1. If you
Okay, you
download the
already
one,
have
I
it.
already
Yeah.
have it.
So if you download the third one,
It will show 2 now.
oh.
Before you go and download the one, I see you did not see 1 in bracket.
So
Then
if
you
you
are,
download ours, it will just show
yeah,
this
it will not
without.
show 1. I think it's worthy pointing out. Somebody will now go and say, Sadio, I downloaded does not have
what
1.
I'm trying to explain, but I didn't get the clear terms.
Eh,
That's
so,
what I'm trying to
so better for you. Okay, so I've clicked, you double click, even though I set mine at single click, but if you double click, it will open it. So this is our protein. Okay, if you open this for the first time, your mouse will be by default in select mode. Can you see? I can't move this protein because my mouse is in select mode. Most times, if I'm working on a project like this, I have my external mouse and I attach to this. Not because this is a new system, not because this is not working, but you external mouse has more options than this. In this, you will need to be converting, unlike external mouse that have many buttons. You can easily, I don't know if I'm getting what I'm saying. So to move this now, I have to right click and change my mouse mode from select to rotate before I could now move this. But if I'm working with mouse, I don't need such conversion. Once I right click and drag it to remove it, left click, I'm selecting, I'm zooming and all that. Okay, so if we rotate this, we'll discover that we'll have, are we together now? So we have varying, we have a lot of things inside here. We have a lot of things. There are some kind of red, red, red. Uh, no, you're wearing glass. Can you see that? Okay, there are some red, red dots there. Uh-huh. Then aside the red dots, there are some um molecules here. The dog. Good afternoon, sir. Yeah, you will as well find some units here. Can you see some compounds here? Can you see that there are quite many here? Okay. Yes, there are some compounds. Look at one here. Look at one here. Can you see them? See another one here. Look at it here. Aside that, also, this protein tends to have two sides. Okay. This and this, where everything that is here is here. However, you might not notice that by mere looking. But when dot there is not working, it's only that switch close to that dot that is working. Yeah. She just left. Yeah. I don't know. He says he's coming, but he carried his bag. Bag, yes. So when you open this, there is an arrow you are seeing here, right? Let's see the arrow. You tap on that arrow. It will open up. By opening up, it will tell you, give you a content of everything that is inside here. Okay? Yeah, what he has there. Dog, good afternoon. Welcome, man. Yeah, welcome. So here you're going to have your ligand group, your protein group, your water, and then heat atoms. Okay? The same thing is what you have here. You have your water, you have your ligand group, and then you have your protein. Okay. This software,
Is what?
the B,
Yeah.
the first B or the last B downwards is always the water. But if you start from up, dog, help us close the door. Uh, The fourth alphabet upwards can always correspond to the protein, which is the highest molecule here. The second one will correspond to the ligand, while the last one will correspond to water. Sometimes you can have this thing to F. Do you get that? You have A, B, C, D to F. By instance, you have this number reach here, then here you have a very, you simply keep removing. That protein is what you must use. You keep removing them until you have just one.
Which is that top one?
Which is that top one? Yeah, usually the A. Just in rare cases you can use B. Okay. Yes, but when you start working now, I'm just showing you this because I've interacted with some of all this. You discover that the literature you might be working with, the active site study they investigated. Remember, you have subunit A, B, C, D. As you're having this A, B, that's the subunit here. Are you getting it? It might be that the person used directly B, okay, not A. Okay, which sometimes is just predisposed to choice. But to me, it's recommendable that you use the A. Okay. But in that case, if you check the A and then the active site configuration of the A is not actually aligning in the literature, you might see arginine 20, and then in your A you are seeing arginine 21. What it means is that in the B you are going to see arginine 20. So it's just a little kind of deviation from the normal, but within the active site, that conserved group, which I mentioned before, will always be there because they are the one responsible for the catalysis. So, but in most cases, what I usually use is my A. Is that alright? So if you use that, it will still have that. Okay, so what do I do here? I don't need most of what is here. First of all, I'll have to reduce this to one unit. So I will remove everything that has B here, right? I'll first of all go with the third one, which is the water molecule. If you want to know that this is water, if you click on this, what is it called? This arrow. Yes, it's going to tell you that what you have here is HOH, which is what? And then it's going to tell you of course, you have many water here. Is that also? According to each of these correspond to this single dot. So you can scroll down and see that there are quite many according to their number. Can you see that this corresponds to all those water there? So I'll highlight this and just hit on it and then click on delete. and it is gone. Is that okay? Then I'll go to the next B, which stood for the ligand. If I click on it, you see as they'll get highlighted, right? They'll become yellow here. Look at it again. If I click on this, they'll turn to yellow. It means they are highlighted. So I can hit on my delete button, and that will go, right? Similarly, I will hit on this, the next B. This next B should be the what? The protein, right? I see highlight, so I'll do what? I'll click on delete button again, and that will be deleted. Is that alright? Then this is what I now have A, right? Corresponding to A water, A ligand, and A molecule protein. Okay.
What if by accidentally
Hmm.
you deleted the one that you don't
You're
want to
not supposed to. Just do Control Z, and then it will come back. Is that alright? However, as you're doing Control D, if I do like this now,
control it?
Control Z. If I do like this, it will recognize it as a step. If I do like this, it will recognize it as a. So when you are doing Control D, Control Z, it will keep. Assuming you deleted this and you kept on rotating this for like 10 times, and you now notice that something was wrong, you want to bring out that one, you just go and do one Control Z. It will not come out. All those shaking you were shaking, it will keep on doing it, going back one after the other until it reaches that place where you deleted the protein. I'm just all this can only come.
Don't touch.
No, you have made, you don't know that you have made mistake, right? You can. I'm just trying to tell you that if you delete something, okay, incidentally that you mentioned, and then you continue your work, I now remember that Control D can bring it back. A single Control D will not bring it back. That's just what I'm trying to say. You keep doing it until it reaches where you deleted it.
You not mentioning it about this thing now.
Heat atom. Okay, heat atom is just telling you about, you can see that here. If you click on it, anything you don't understand, you just highlight it. It will tell you what it means. Heat atom is aside your protein, your this thing. You can delete all of them at once. Okay, if you wish, but I decide to show you one after the other so that you see how.
You can just hit on it directly.
Yes, once you hit on it and click on the it will go. Okay, so we have deleted this B before, so I'll take it off. Okay, so this is what we have, right? I will, yeah, I will go ahead and delete my water. Okay, right? There's no water. This water, who can tell us what this water stands for? This water we just deleted. We believe it's water, right? Why is there water in this structure? We not even wonder. That's the water of crystallization, right? You can't do crystallography without water crystallization. Do you understand that? So that's why it's there. Okay. That well taken. So this is the ligand. Then if we look at here now, there are many ligands here, right? And then there is even a cofactor. This is a cofactor. Can you see that? There's something here. No, not dot. If I open up this ligand group here, there is a dot there, but it's green. Can you see that? This is it here. Can you see that? There is a dot here that is green. This is what we call the cofactor. Okay, some of these metallic units that some like calcium, magnesium, some proteins use this in the course of their catalysis. So while elucidating them, you still find them in the structure. However, you take off all that. Okay, just to get your protein safe from clashing with any kind of interaction, except your literature indicates that is necessary. Right? So that is that. So what I usually do here is to remove the protein by just highlighting and unchecking it there. Okay, if I uncheck this bracket here, the protein will go. Is that not so? So I want to uncheck the protein so that I can study the ligand. Okay, as you can see, there are many ligands here. These are lipids. Remember, this protein is all about lipid metabolism, arachidonic acid metabolism. Is that not so? It converts arachidonic acid to prostaglandin G2, which is the oxidized form of prostaglandin, which will reduce to the produced from prostaglandin. Sensation of pain is experienced. For some of us that have studied mechanism of inflammation, this is how it comes about. Okay, so if we look now, there are um, many groups here, right? Look at that one I was talking about before. Can we see that? So these are many of them. These ones here, okay, let's bring out this back. You discover that these ones here that are binding here are quite outside. Can you see them? Outside the protein, can you see them? All these ones are outside the protein. Okay, even this one I try to enter inside. But something like this one is inside the protein. You see this one is reside that will tell you that, okay, it's not like magic I'm showing you. That one is inside. Then another one that is inside again is this one. This one here. This one is inside as well. Okay. Then how did I know that these two are inside? This is what we call, okay, let me zoom this. I'll go to mode and then I'll go to zoom. Okay, let me change this back to the base. You can see the stress because I don't have mouse, right? Look at the heme group. There is what we call heme group in heme group in peroxidase catalyzing reactions. We call the heme group. This is exactly how the heme group look like. Okay, they have an ion center. Some of us that remember that, you remember what I'm talking about. Yeah, so look at the ion center, look at the heme group. Wherever you see this, it's one of the feature of a peroxidase active site. Are you getting that? Yeah, it's usually made use of by peroxidase during the mechanism of the catalysis. Then another one which is inside is this one I mentioned here. This one here. Can you see that? This is the the drug, naproxen drug that was used to target this protein.
Which one?
This
This.
particular one inside here. Okay, then how did I know that this one is inside, is the drug? Okay, let me say that I know about this because I saw the heme group, right? Then how did I know that this one? Literature told me that the active site of this contain the one I can remember very well is arginine 102. Okay, the ligand binding site catalyzed by cyclooxygenase 2, which is this protein, has two sides. This is the protein, right? The literature told me that it has two sides. One side is the membrane binding domain, which is the first point of binding for that reaction to occur. That's the first point where arachidonic acid binds, is in the membrane binding domain. And the enzyme that catalyzes that particular domain is the cyclooxygenase. I get it. That's the first point of catalysis there. Before it now moves to the peroxidase site, where peroxidase will now play a role there. Peroxidase is an enzyme. Okay. So here now, I know that that active site has arginine 102. What do I do? I'll simply search arginine 102 here, and it will show itself, and I will know if it's here or even here.
Okay.
Do you understand what I mean now? So what do? I will right, okay, I don't even need to right click here first. I'll simply come to this protein. This is A, the protein, right? We agreed about that, right? So what do? I will go and open up this arrow. If I click on the arrow, what do you think will happen to the protein? What do you think will show?
The content.
The amino acids, right? It's going to show the amino acid, which arginine 102 is going to be part of it, right? Okay, so I'll simply open up where the protein, I'll open it up. Okay, and when I do, I will scroll down to, it started with alanine 33. So I will scroll down to 102. I hope I got that very well. That's arginine 102, right? And 103. Arginine 120, sorry, not 102. Arginine 120. It's almost getting off my mind. It's arginine 120, but I remember it's arginine. Is that okay? So arginine what? 120. I told us I got that from literature, right? Is the ligand binding amino acid. If ligand comes into this pocket, it will go and bind to it before the neighboring amino acid can now start electron abstraction and electron transfer. Is that all right? Okay, so I'll simply highlight on the arginine. If I highlight on it, it will show here. Okay, let me click outside. If I highlight on this by clicking on it, it will show here.
Yellow.
That is this yellow. It won't just show. I will now come outside and right click. And then I will change the display style by clicking on this display style. I will change that highlighted one, it's still highlighted, right? I will change it to ball and stick. I'll change it to what? Ball and stick. And then I'll hit on what? Apply. And then I'll hit on what? Okay. Look at what has happened here. Can you notice what has happened here? I've clicked outside now. This is the arginine now. How many of us have seen an arginine before? Do you remember the structure of arginine? Even you are supposed to
I
remember.
don't remember. So
This is arginine, and remember arginine, basic structure of amino acid is the carboxylic end, the amino end, and then the carbon end, right? And then hydrogen. The simplest, which is glycine, has one carbon, a central carbon, amino group, nitrogen group, hydrogen group, and then I think a carbon atom. Is that not so? The same thing is what happens here. You discover that this arginine, aside that end that has the amino backbone, the other end it has two nitrogen groups. Arginine is there, lysine is there. While something like glutamate, aspartate, these are what we call the positive group, while the arginine, lysine are the negative group. Some of us know they read, that is why. Is that which one is not
those things I've forgotten them.
This is protein. You know classes of protein. You know amino acid. There are 20 essential amino acids. You have the inside that 20 there are neutral amino acids. There are positive amino. There is positive and negative. They will deny that they are not healthy.
No, I'm trying to remember if I have seen something like that before or heard about it before.
Hey, even in primary school they taught something like that. Okay. Okay, so if you see now, this is the arginine 120 here. Okay, this is the arginine 120. The ligand here is very close to here. Is in this pocket. You won't see this view here as in the pocket, but there's actually a pocket inside here where this thing is binding. If you view this thing in pine mode, you see that there's a pocket. Okay. Let's quickly open our pine mode. Let me show us. Okay, you just type pine mode here, and then you hit on this on your search. Okay, yeah, sure. No, it's offline. It's an offline too.
That's the one installed.
Yeah, that's the one I installed. If I open, if I open, let me go to download. Let me go to this, and then I'll go to this. If I load this, let me put it in. This is the protein. I'm going to show us this later. If I put this in surface view, okay, this is it in surface view. Let me show us that this is the heme group. Can we see the heme group? This is the heme group. Okay, let me see. Okay, look at what I'll do. Let me put this in red color. Okay, then this is the heme group. Let me put the heme group in green color. Can you see that heme group I was pointing out towards that time? Can we see that this is the heme group for this particular unit? Look at that middle. We are still working with the original distance, so the two, everything is still here. This is the heme group of
This is the unclaimed protein now.
This is the unclaimed one. Look at the second heme group here. Look at it here. I'll put it in yellow as well. Look at it here. Then let me look for that ligand, right? Okay, which I was trying to show us that is in a pocket. Let me show us where it is. Look at it inside. Is it the? It's because this one is still yet unclean. It should be somewhere close to.
Inside this file, can you clean it?
Yeah, you can. You can. But you see that inside. I'm supposed to find it here. I'm supposed to find it here. Okay, look at what I will do. I will save this quickly as yes, so that I just went to open time and go to. We'll get to that. Okay, I'll save this as clean protein. Clean protein. Okay, and then I'll hit on. You can see that file type, right? It must be saved as what? So don't be in a haste to just click save. And so I'll save this. Then I can now come to buy more now. And then under action, I'll delete everything. I can go here and open this now. And this is it. Okay, I did not even remove. Was I saving? Okay, those ligands, some of them were still there.
We are not done.
Okay, we are not even done. Okay. I thought as much. Look at that ligand inside. Look at that ligand inside.
The green ball is the cofactor.
Yeah, the green ball is it. Look at the ligand here. That is this one.
This one.
Yeah, this one. This one I just highlighted. Let's put it in surface view so that we can see that. Okay, this is the reason why we're not able to find it. Can you see it inside here? Can you see it inside there?
It's green now. This green one.
Is it green? This one I'm touching here. Look at it inside. There's something inside there. Can you see that? Yes, that is actually. I think there should be another. This is where he entered from. Not this. Look at it inside here. That is this group. Let me change it to red. So if I turn like this, you see that red portion. Can you see it? So this is where I know there should be an entrance. So if you target that amino acid, it will enter through here inside that. Then that entrance there has a link, a point link with the heme group, because from that pocket there, the product will move to the peroxidase group where the oxidized prostaglandin will be reduced to normal prostaglandin. Now in the seed pain. I don't know if we are getting it. So I'm just trying to show us that these things are in pockets. They are always pocket like a hole. Okay, but in this two, we're not able to see anything like that here. Okay, if you can see now, you just look like.
You said you wanted to show us where the arginine binds to the ligand in this line.
Okay, why we went to Pymol was to show us how this thing here is actually in a pocket, which we might not be able to see here. Okay, and then remember we started with trying to locate whether this is actually in a pocket or not. That was why we searched for arginine 120, right? And we discovered that the arginine 120 is in the same pocket as this particular protein, this particular ligand. So I can now go ahead to delete every other
They are in the same pocket, not like it binds.
No, it's not binding to it, it's in the same pocket. However, it might be binding to it, but you will see it. You will see it like this. I will show us. You won't see it like this. But before we do that, you have to remove every other forms of binding here. If you want to investigate a particular binding, remove every other so that these two will understand that you just want to investigate that singular. If I hit, for instance, like this now, if I hit the 2D view, which is supposed to show me the ligand protein interaction, look at what it will give me. It will give me this. Out of the many that are there, it just gave me this, which this is just one of the atom here. I don't even know which one. Are you getting it? So you won't just take this to be your ligand. So what do? You take off every other ligand here. Sometimes it will even give you one funny structure like that. So what do? Let me zoom this back. Um, okay, so we can now close this. Remember, I've been able to show us that arginine. We are still coming to something there. So what do? I'll take off this protein, just highlight it, and then put this back to highlight mode or select mode. I want to delete every other thing aside the main ligand, right? Including this heme group. I already know heme group is in the peroxidase end. So what do? I will just highlight this way and hit edit. You can see the two cofactor here, right? So I will still delete this. Yes, I don't need them. They are gone. So this is okay. I can change this to rotate now. So this is basically the only ligand I have here, right? So I can now bring back my protein. Can you see that? This is the ligand inside there. This is, remember now, the arginine. Okay, let's now hit our 2D view now. Can you see? This is the ligand now interacting with what?
Okay.
The arginine what?
120.
Do you understand? This is the native ligand interacting with arginine 120. That is why, at the course of this, I'm going to tell us that one of the way you validate how well your research result was is try to compare what you got to number one, the co-crystallized ligand of this protein. That co-crystallized is like the native ligand. It could be a drug, it could be its substrate. For instance, if you work with native acetylcholinesterase, the substrate of acetylcholinesterase is acetylcholine, right? If you co-crystallize it with the native form, visualizing acetylcholine inside acetylcholinesterase will give you an idea of what or how ligand interacts with acetylcholinesterase. The same thing is what I'm showing us here. We have seen the ligand, the arginine 120 now, interacting with the ligand, right? If you drop your own and your own could not interact with arginine 120, it tells you that there is a problem. Okay, that is why in this software by default will generate up to 9 different conformations of your ligand inside the active site. By that I mean that when you send your ligand into an active site, the ligand keeps interacting with the units or residues of the active site into 9 different conformations. It will stay like this, it will stay like this, it will stay like this until 9. By essence, later you're supposed to visualize out of these which one best. Remember, if you still remember what we did the other day, we discovered that one of the essence of talking is to get or predict a better binding pose. I don't know if you remember that. Binding pose or conformation of your ligand within the active site. So that you can actually see, oh, this ligand can actually interact with this protein. And this is how it does. So you can see that this native ligand or inhibitor of this protein was found you see this one, you see this tyrosine 355, and there is another one. This one is for electron abstraction. This one is for binding from literature. This is for binding. I think I will find that literature and send to us so that we'll study this thing I'm talking about. And I hope I can find it. I hope I can find it. Okay. So you will discover that this is for binding. This one close by. Once this binds, this will initiate electron what? Abstraction. Catalysis is all about movement of electrons. And also it will abstract from the substrate and transfer to the flow, and then the substrate will now become a folder. Just that. So that is exactly what is happening. Are we getting that? Don't worry, we're rounding off almost. Okay, so this is what we now have. So I've got this. Most time what I do, I will save this. Okay, I'll go to save as and save this as um, maybe internal ligand. Internal ligand. Normally, you say 2D internal ligand. It means the two-dimensional structure of the word. Internal is already an image view. Are you getting it? This is to serve as a guide. When you start working, after docking, you're supposed to check, compare this to what you have. If you don't have this, remember there's possibility that you have the first result. This arginine 120 will be here. You won't be interacting with your ligand. You keep screening your ligand until you find one that is interacting with it. That one that is interacting with it had a better pose than the rest that is not interacting with it. Is there anything at all?
Well, inside here, read this one that is the ligand.
This is the ligand. Let me check. Okay, let's save this. I'll say okay. Okay, let me change this so that we will. This is what we call the atom. So it can be in ball and stick. This is the ligand.
Okay, this one's in boss.
Yeah, this is the one in balls. Of course, this is the amino acid. Amino acid will be ligand.
So that we know the one that is
Yeah, this is the amino acid. And then these are the OH group or the oxygen. We just call them the oxygen group. This is actually oxygen. Why this is what? OH, but this is oxygen. Who can tell me the difference between these two?
This oxygen, there is double bond.
There is double bond, when this is just that. So if you go to the structure of that particular compound, you're going to find something like that. So I've saved this. This is the compound, and this is the amino acid. I'll still show us about customization later. So if I zoom this further, let me show us what we just noticed there now. Okay, I'll go to my display mode and then I will zoom it. If I zoom that and change this to rotate, you discover that look at the compound, look at the ligand, right? See the, yeah, see the oxygen with double bond. Can you see that? See the okay, let me close this and then maybe I could zoom further for our sister that has 20 eyes. Okay, so this is what I have. Can you see the double bond oxygen? Can you see that? See the single which we saw in that. Then aside that, look at this, look at this. This is how I know which of this group is interacting with this. Remember, this is the amino, this is the arginine, right? Which at the course of this, we are going to target this arginine. We are not just targeting arginine, we are targeting a functional group in arginine. I cannot go and target this carbon here and ask this group to come and interact with this carbon. You know it will just be a mess up, right? So in targeting, you target the what? The functional groups that are reactive. If oxygen is not there, nitrogen will be there. Nitrogen is not there, sulfur will be there. These are the three you can target. Hydrogen is not referred to as beta functional group. Is that alright? Okay, so what do we do now if you zoom that closely? Okay, you're going somewhere. Okay, so how do you want to go about it? I'm not sure these people have water here, except you can walk down to cheat it.
Hey,
just keep it.
no, don't come back.
No.
You know where it is now.
It is now, you know.
Eh. So if you look at this now, you discover that the closest to this group is this.
So which group?
So this thing is this. When compared to the rest, I don't know how I'm to show this so that you see. These two are actually nitrogen. This is nitrogen. This is nitrogen. This is nitrogen. Right? But these two look farther than this,
By
right?
saying
Yes, by saying.
it's not the color, they have three colors.
Uh-huh.
Not by
Same color. You say by seeing it or tracing it, are you going to make
it sees all from
I
Okay, sulfur is always in yellow color by default. Hmm? Sulfur is in yellow color. Carbon is in ash color. Hydrogen is in whitish color. Nitrogen is in green. Oxygen is in red by default.
So this one, nitrogen, now is in blue
Yeah, this is
by
nitrogen in
default.
blue by default.
Mhm.
Okay. Then carbon is in ash by default. Okay, so what I'm not looking at is this nitrogen. That's the closest to this bond. If you notice the 2D, there's actually this nitrogen that is interacting with that, but you won't be able to see it there because it's interacting with the whole group here as a group. But this will come into play because if I tap on this, something will change. A write up will come out here. If I tap on this like this, a write up comes out here. Okay, look at this. Something changed there, right? If I tap on this, something changed, right? And I'll be looking at this number, be looking at this NE, see if something is changing there.
NH2, NH1.
Are you seeing that? Do you see that now? What it means is that these amino acids as well, these functional groups here, these nitrogen groups, have their individual coordinates. If you want to target arginine, you will as well consider which group in arginine are you targeting. That is the site specific now. Oh, you know the time. Okay, okay, okay. Let's. I think we can end it here, right? Where we take our questions, right? How about that?
Mhm. Yes, sir.
Okay, so we can end it here. Uh,